Good afternoon, everyone. Welcome to my class of human genetics this fall semester. Although we will not have a face-to-face session due to the pandemic, I hope the online sessions will provide you flexibility and make reviewing of the lectures much easier than before. Today is our first lecture. We will mainly go through the whole setting of this course and introduce some fundamental knowledge. At the end, I would like to ask you to do a small test online. These slides list the career possibilities of a person who studies biology. Although some people may not work on biology-related work, such as banking or finance or accountant, most people will enter health-related field. A lot of you will go to graduate school, medical school, or dental school, and will become university faculty physicians or dentists in the future. Taking the human genetic course will help you better prepare for admission exams and understand your own body at molecular level much better. So moreover, I hope every student can benefit from the course more or less. So this is the overall goal and purpose of this course. Specifically, here is the course goals. The first goal is to strengthen the fundamental biology concepts and the principles about human genetics. Many of you have taken genetics, which already covers some of the knowledge here. So I'll go through this part relatively quickly, and we'll focus on problem-solving strategy. We will go through example questions together in the class. The second goal is to help you understand the molecular basis of human variation and diseases. We will go through the molecular events of how babies are formed, why everybody looks different, and how genetic diseases will occur. If you plan to go to graduate school or medical school, this part will be very relevant. Meanwhile, for the third goal, I'll introduce modern biotechnologies to you, including the principles of these techniques, applications, as well as cost and limitations of these technologies. If you plan to work in a pharmaceutical or biotech company after graduation, this part will be of interest to you. Okay, a little bit background information about me. I have been studying biology, biochemistry, and molecular biology in my whole life. I obtained a bachelor's degree and master's degree from one of the best teaching universities in China. Later, I came to US and earned a PhD degree from UNC Chapel Hill. After that, I moved to Houston and worked as a postdoc and then junior faculty at Baylor College of Medicine. I stayed at Baylor for about 14 years before moved to UH as an associate professor about 2 years ago. So this class is the third class I teach at UH. My lab mainly work on gene regulation, meaning transcription. I study HIV and sexual hormone-related cancers, such as breast cancer and prostate cancer. Basically, we try to understand the transcriptional regulation of the key molecules in these diseases and further design targeted therapies. I will also integrate some of the research aspects into this class and show you how scientists think, design, perform experiments, and report their discoveries. Here shows the recommended textbook for this class. So the current version was published in 2015, actually quite a while ago. Although some of the materials are a little outdated, this book is very easy to read and gives you a nice introduction on the basic concepts. We'll follow the chapters of this book and integrate the new materials as necessary in my lectures. Hopefully, a new version of this book can be available soon. I estimate that about 2/3 of my lecture is going to be based on this book. It has a great price if you choose to rent this book in an electronic form. Here comes my recommendations on how to use the book. While you read the book, I would like you to identify the key points, which are usually shown in my lecture slides as well. Pay particular attention to the glossary and definition on the margin. Figures and tables are usually important. Try to understand them well. So for important concepts, try to remember them as well. When you, as I read here, not only understand it, but memorize those important figures, tables, as well as the contents. Now let's go through the structure of this course. I'll divide the course into several parts based on the exam dates. This is showing the first part that will mainly focus on the classical human genetics and cell biology, all right? So we have 7 lectures and 1 exam will be based on the content from all 7 lectures. I'll post a video link of 7 lectures in the announcement the same way as this lecture, so you can view these lectures at your convenience. However, for all of the exams, you need to complete the online exam in a designated time. For instance, exam 1 needs to be completed during the time 4 to 5:30 p.m. on Monday, September 31st. For the second part, we will focus on the molecular events related to human genetics. The second exam will only cover lectures from 8 to 13, the second part. For the third part, we will introduce newly developed biotechnologies, as well as how they can be used for treating diseases. We will supplement a lot of new information, such as immune therapy for cancer treatment. For those topics, please follow my slides for study, okay? Besides the regular lectures, I also prepared 2 research papers to introduce how scientists solve questions and report their discoveries. Following these 2 lectures, we will have another online exam. Again, this is an open book exam, and it has to be completed on December the 14th, 4 to 5:30 online. So therefore, the online attendance of all the exam is required. Okay, so this slide shows how I handle grading, all right? So the 3 exams on regular lectures counts 60% of final grades, with each exam 20%. The last exam on journal papers accounts for 10%. We will also have a group project that counts for 30%. I'll give you an introduction of this group project in the next few slides. You also have a chance to earn 4 extra bonus points toward the final grade for those of them who attend the live online sessions of group project. If you also fill out the grading form, which I'm going to distribute before the online sessions and return to me after the lecture, then you will earn 1 bonus point for that session. So altogether, they are up to 4 points in total. This slide shows the grading scale, so it's pretty easy to understand. So I'm not going to go through in details. You may want to know the performance of prior students in this class. This was the statistics from my first class. I had 37 students in that class, and 28 students got B- and above. But I still have like 9 students had a C+ and below, so which was not very satisfying. Since this semester, all lectures will be delivered online, it is particularly important for you to set up a goal and put effort on it. And you'll get rewarded at the end in that way. All exams have been scheduled on the designated dates as shown on this slide, okay? They are all made of multiple choice questions and open book. Importantly, all exams are timed. Exams on regular lectures have 50 questions, and you have 40 minutes to complete. When the time ends, the exam will be automatically submitted. You have also I designed a sample test at the end of this lecture, so you can get familiar on how these exams will be handled, and you can see the format of those questions as well. Please make sure you complete the test. The score of those sample tests does not count toward your course grade, so it is only for practice purpose, okay? Now I would like to give you some details about the group project. It weighs exactly 30% of your final course grade, and each group will work on one genetic disease. We'll have about 3 to 4 students per group, depending on how many students will stay in this course. I'll notify the number a little bit later. So you will be asked to form the group yourself, but for anyone who does not form the group voluntarily, I'll assign groups for them. The project can be done in the form of oral presentation, or it can be a movie similar to some YouTube movies I'll show in class, or any combination. Use any creativity you have as long as you stick on the goal, and that is for the audience to understand the disease better. So you are required to send me the link to your video a day before your presentation time so that I have time to put everything in order for the class, okay? So the video should be approximately about 12 minutes. And then a short question and answer session will be handled after each presentation or video. How well the group will handle questions will be also counted as part of their grades. The students' audience will grade together with me. And also the scoring rubrics will be available in late September. All right, so this is a timeline about the group project. On the top is what you here for the students, what you need to do as the timeline come. And on the bottom is what I need to provide to you. So I encourage you print out this page so you don't miss these deadlines. I actually print out this page as well. So all project related information can be found in a folder named the presentation or video project on the content folder in Blackboard. Regarding to that, I would like to show you where you can find those course related materials in Blackboard. The most important location is the announcement. All of my notifications, including like each lecture links, will be posted here. Other course related materials can be found in this content folder. So I have several folders that are pretty self-explanatory in this content folder, and you should be able to locate them easily, all right? So the lecture slides is going to show in PDF form. So I'm going to deposit or upload it to Blackboard after I uploaded the online lectures. So you will be able to find them and easier for you to go through the course materials. This slide is a summary of course delivery methods. We already went through most of these in previous slides. I'm not going to repeat anymore. But I would like to mention that we also have optional Q&A available as a form of Zoom meeting on Wednesdays, starting from 4 to 5:30. You can find the schedule in a syllabus, the last page, okay? So bring your questions in for those sessions. I will also post the Zoom meeting ID in announcement prior to those meetings. So group presentations, exams, and the Q&A sessions will not be recorded. So I have always been asked about the, how about if I cannot attend the exams due to a time conflict? Can I get a makeup exam? Okay, so generally speaking, unless you have an emergency that prevents you from attending exams, you should always try to arrange the time. since you already knew the course schedule when you enrolled the class. Otherwise, you are risking your grade. But of course, in case a real emergency hinders you from taking the exams, you need to let me know as soon as possible. So here lists the conditions that qualifies for a makeup exam, all right? This is the last slide of course introduction. Based on my limited experience teaching students, I summarized a few things that are the keys to score high in my class. When you are viewing my lectures, I hope you are not just learning passively, okay? Instead, always try to make connections to any knowledge you already learned. You will raise questions in this way. The more you think, the smarter you are going to be. Try to stand high and focus on the big picture first. If you would like to know more about something, then you go to the details. Some students like to play the video in a faster pace. I do not recommend that, okay? Because then you don't have time to think, probably not even have time to understand my English. Under that condition, the knowledge you get will be forgotten easily, all right? So before the exams, also make sure you review my slides frequently and memorize the concept I emphasized in class. The exam, the time is limited, okay? So if you're not familiar with the course content, for sure you will not have enough time to go through the slides and find the answers during the exam. So be aware of that. Okay, now I would like to play a YouTube video on introduction to our human genome to get the course started.
Humans around the world have much in common, but also enormous diversity. Some of the differences between each of us come from our environment and life experiences, but our DNA plays an important role in determining our appearance, our traits, and our health. There are thousands of genes in the human genome. Sequence changes in individual genes can determine if we have freckles, can digest lactose, have wet or dry earwax, are red-green colorblind, or are likely to have blue eyes, or think broccoli tastes bitter. Individual genes can also determine if we will develop sickle cell anemia, cystic fibrosis, or Huntington's disease. Multiple genes act together with our environment to determine our hair and skin color, our height, our weight, our blood pressure, and our risk of developing type 2 diabetes, depression, cancer, some autoimmune disorders, and many other conditions. In spite of all these potential differences, humans are 99.9% genetically identical. How is it possible that we are all so similar and yet so different? Let's zoom in to the smallest genetic unit, a single nucleotide of deoxyribonucleic acid, or DNA. A DNA nucleotide is composed of sugar and phosphate groups, and one of 4 nitrogenous bases: adenine, thymine, guanine, and cytosine, annotated in shorthand as A, T, G, and C. The sugar and phosphate groups form the DNA structural backbone, allowing nucleotides to concatenate into a long single strand of DNA, while the bases determine DNA sequence. The chemical properties of DNA allow bonds to form between the bases in order to create a double strand, with 2 hydrogen bonds pairing A and T, and 3 pairing C and G. Though different types of human cells can be very different in appearance and function, they contain the same genome, which consists of about 3 gigabases, or 3 billion base pairs, of DNA. All the DNA in the cell would be about 2 meters in length if it were stretched out, and must be condensed down to fit into cells as small as 10 micrometers across. The DNA is first coiled into its canonical helix structure, and then wrapped around histone proteins to form a DNA protein structure called a nucleosome. These nucleosomes can be further wound and coiled together to create a compact structure that fits into the nucleus. During cell division, The DNA is organized into tightly wound chromosomes, 46 in total, with 23 coming from each parent. These chromosomes can be easily and accurately separated during cell division, guaranteeing that each new cell contains an exact copy of DNA. Outside of cell division, the DNA is decondensed in the nucleus, allowing greater accessibility. The transcriptional machinery regulates expression of the approximately 20,000 genes, which, although they correspond to less than 2% of all genomic DNA, encode all the proteins necessary to build and run a human cell. So to return to the original question, how is it possible for all of human diversity to exist when we are 99.9% genetically similar? It is important to remember that the 0.1% of DNA that varies, on average, between each of us actually corresponds to about 3 million differences across the genome, with 20,000 of them, on average, falling into protein-coding genes. Although that equals approximately 1 difference per gene, in reality, these differences are not evenly distributed across the coding regions. Differences in DNA sequence are called variants, and those affecting a single position are called single nucleotide variants, or SNVs. Common SNVs that occur in more than 1% of a population are called single nucleotide polymorphisms, or SNPs. SNPs, along with larger scale sequence changes like deletions, duplications, and rearrangements, create all the richness of human genetic diversity at the population level. This raises many questions on an individual level as well. How does DNA determine our traits? How can we understand what a gene does and how variants in that gene might affect our lives? What can our DNA tell us about our risk and our loved ones' risk of disease? How can information from our genomes improve our medical care? Understanding genetics allows us to apply the concepts of heritability and genetic variation to questions of human health and disease in our world today.
All right. How does genetic information affect our real lives? So I would like to share you a real story. I will just watch the video, right? So I read this story on Father's Day last year, and it was quite impressive. And I thought this is a good example to integrate into this course. Okay, so Dr. Stuart Scriber is a scientist at Harvard, and co-founder of the Broad Institute is a 4,000-person biomedical center. They're seeking to propel the understanding and treatment of disease. Okay, he wrote what he experienced in this story. So Dr. Scriber was brought up in a family with his father very restricted to him. In the worst of times, his father beat him so badly that he could be hospitalized. But in contrast, his mom was very sweet and caring. This is some background, okay? So in 2017, Dr. Scriber and his older brother, Tommy, did a genetic testing using a kit named 23andMe, okay? So they trying to find out their risk alleles for Alzheimer's disease, which had taken their mother's life. So the result was very surprising. As a scientist who had been working on genetics for many years, so Dr. Scriber immediately realized his biological father was not the man who raised him. Just once he saw the result from 23andMe. So Tommy and him shared only the 25% of DNA identity of half siblings, so not the expected 50%. And their father contributed the Y chromosome different. All right. So this is a photo of Dr. Scriber's parents. So that's his dad, that's his mom. So in my opinion, although Dr. Scriber's father was not his biological father, they do look like each other, right? So before we get into the story, I first want to introduce this genetic testing kit called 23andMe. Okay, so 23andMe is a direct-to-consumer genetic testing service. So using customers' saliva sample, The lab does a genetic analysis and generate reports relating to the customer's ancestry and genetic predisposition to health-related topics. You can do health as well. So in 2008, this business was named as Invention of the Year by the Times Magazine. So this image is a screenshot from this company's website. You can find better prices probably in some other vendors such as Walmart when this kit is on sale. So I think the typical testing is recommended is $199 from company's website. But how does this kit work? So when you receive the kit, you will find a tube in this box that you use to collect saliva, which will be later sent to the labs. The company will perform single nucleotide polymorphism genotyping. It's also called SNP genotyping. We just learned that, right? The sequence of your SNPs will be used to compare with a big reference SNP database, and the corresponding phenotypes can be deciphered. So if you are interested in knowing more about how this works, check out this website, this YouTube link, which give a really nice introduction. So this slide shows a sample report of 23andMe results in ancestry. It tells you how many percentage of genetic information you got from different lineages. Here you see this guy actually has different lineages, have different percentile as well. So also the risk factor of different diseases are included in the report. Here is an example. This was the main reason why Dr. Schaber and his brother did a test to see the risk for Alzheimer's disease, because as here you can see it has all these different risk factors, also including Alzheimer's disease here. Okay, let's continue the story. After Dr. Schaber found his father was not his biological father, he looked into other databases. Within 2 months of searching public and private DNA ancestry database, he performed the computing, construction, and the connecting the family trees up and down multiple generations. We're going to learn those things later on. So Dr. Schaber eventually discovered the identity of his biological father, who had lived within walking distance of his mother's home and named Joe. So that's here, this is him, this is mother, and this is really his biological father, okay? He even found a series of paired age-matched photographs of Joe and him, and they shared the same elbows, eyes, noses, nose, ears, chins, and they were both bald, also at age 35. So unfortunately, Joe passed away years ago, and Dr. Schaber was able to find many new family connections, such as half-brothers and half-sisters, okay? So make the story more interesting, he applied the same skills. He revealed a DNA-guided path to his mother's actual father, named Dolly, because initially Dione, you know, it's not known to him. Then this is actually a second new family. He learned that his biological grandfather, Dione, is a moonshiner and was purportedly on the run after killing a man in Mississippi in the old days, and then met with his grandmother. Actually, she was a prostitute and gave birth to his mother, all right? As a matter of fact, his mother did not even know this. So people in the blue shape are the ones Dr. Schaber initially knew, and people outside were the new relative he found by this genetic searching. So this is what a surprise when I saw this, story. But after reading this story, I later encouraged my son to do this 23andMe test. So, but we unfortunately didn't find such an interesting story from my son's test. But we indeed identified several like fourth and fifth cousins in the United States who had also done 23andMe before. So because this is such a database. Actually, because 23andMe has so many people's genetic information, it is quite easy for them to do correlation studies. For instance, recently in June of this year, 23andMe published results from a study that showed the people with type O blood type, okay, type O, I think is a pretty common type, the most common type in all the populations. They may be at a lower risk of catching COVID-19. So out of more than 350,000 participants, those with type O blood were about 9 to 18% less likely to contract the virus. So, but then those who had been exposed were 13 to 26% less likely to test positive. So you probably heard about this finding before, right? So genetics are pretty powerful with the population studies. Okay, so now let's introduce some basic concept about genetics. I believe everyone knows that DNA is the genetic material, right? And it exists in the cells as a form of chromatin or chromosomes, depending on the stage of cell cycle. The basic unit of chromosome is called a nucleosome, all right? So which is composed of histones and the DNA. DNA has a double helix structure, and the gene is a region of DNA that encodes a function, meaning genes can be described and making proteins. How many genes exist in human genome? So we just watched the movie. So approximately 20,000 genes. So what is the percentage of genome do this gene occupy? Remember, also in the video, it's less than 2%. So these numbers are important and you should remember them to save your time in the exams, okay? Here comes the central slide of this course. That is the central dogma, okay? So everything we were learning in this class can be included in this slide, which I'll show the complete slide later on. So we learned that the gene is a piece of DNA that can encode a protein, right? And the protein is the effector that really function in our body. For instance, we need to make enzyme to digest food. We need the hemoglobin to carry oxygen. And they are all proteins, okay? But the DNA cannot produce protein directly. It needs a molecule in the middle as an intermediate. So what is that? So that's RNA, right? So DNA is double helix, double strand, but RNA is a single strand, right? The process of making RNA from DNA is called transcription, right? And then the process of making RNA into protein is called translation, all right? So when the cells divide, they need to make another copy of genomic DNA, right? So that process is called DNA replication. When cells are infected with RNA viruses such as HIV or COVID-19, so those viruses carry reverse transcriptase that can generate DNA from using their RNA as a template, okay? So, and the reverse process that is called a reverse transcription, okay? So it is also important to remember these events all occur in cells. DNA replication and the DNA transcription occur in the nucleus, as genomic DNA only stays in nucleus. But in contrast, translation happens in the cytoplasm, as ribosomes are only assembled and function in cytoplasm. So these locations and what they do, you know, the functions and then what material or what other molecules, you have to remember this. This is a central slide of this course. And I'll come to this dogma again and again because this is so important, okay? Since the ultimate purpose of DNA is to produce proteins, then think about what proteins are the most important to maintain the function in our cells. So this picture from Wikipedia summarizes different categories of proteins that are encoded by human genes, all of them. Can you find the classified group with the largest number of genes? Okay, so this group has more than 2,000 genes, and they actually are important. Yes. So you find it, so the transcription factors are the largest group, see? It accounts for more than 10%, 12% over here it shows. They are the key proteins that control transcription. That's the reason why I call transcription factors. We will talk a lot on transcription factors in our future lectures, all right? I just mentioned that genomic DNA only stay in nucleus. Now let's take a look at the structure of the cell nucleus, okay? So the picture shown here, actually the human blood lymphocytes. And you can see those two human blood lymphocytes that usually contain a large nuclei shown at the purple here. But in contrast, the red blood cells do not have nuclei. That's actually the only cell types that do not have a nuclei in human body, okay? That's the blood cells, red blood cells. And then with the electron microscope, you can see more detailed structure inside the nucleus shown here, all right? So it has light material and dark material inside the nucleus. And these both structure actually contains DNA as the form of chromatin. Scientists name this light chromatin as, in this light region called the euchromatin. And the dark chromatin as a heterochromatin. The heterochromatin usually located in the perinuclear regions. That's basically the regions surrounding the nuclear membrane, okay? It's called the perinuclear region. And they are tightly packed with chromatin and enriched in repetitive non-coding DNA. There are very few genes located in heterochromatin region. But in contrast to that, the euchromatin, okay, so their structures are more open. So this is lighted pack, and they're enriched in genes. As a matter of fact, about 92% of a human genome are located in the euchromatin region. Okay, and there's also one or more condensed region here in nucleus named the nucleolus. This is a non-membrane structure, and one major function of nucleolus is to produce ribosomal RNA, okay? So therefore, we say nucleolus is involved in ribosome biogenesis. Recent years, scientists found that nucleolus also participates in gene transcription and RNA splicing. But in many cancer cells, the nucleoli are much larger and visible than normal cells because the cancer cells need more proteins to proliferate, right? So this is actually one of the characteristics of malignancy. So these slides will test some of your knowledge on medicine. Do you know that what causes death in the U.S.? Interesting, the major killer is here, showing this is listed different disease, what is the leading cause of death, okay? So the major killer is the heart disease. It kills more people than all type of the cancer add up together. So cancer is the second leading cause in the United States. So if you become a cardiovascular doctor in the future, you will never worry about the number of patients because there will be so many, right? Because it's the first leading cause of death. The third cause is a heart gas. Yeah, it's accidents, okay? So please drive safely and avoid taking risk that may take your life, okay? So it happens much frequent than what we thought. The fourth is chronic lower respiratory disease, which includes chronic bronchitis, asthma, and pulmonary hypertension, those type of disease. And the fifth one is the stroke. Stroke is a sudden interruption in the blood supply of the brain. And then the 6 to 10s cause Alzheimer's disease, diabetes, and flu, and pneumonia or kidney disease, and even suicide can be listed as the number 10. That's actually a lot. So then you probably will ask, how about COVID-19? Does it list here? So that is a good question. So indeed, this year is very unique. COVID-19 becomes the third killer in the US this year. So, but you can see that heart disease and cancer are still much worse than COVID-19, and they keep in the first and the second positions as usual, all right? Okay, lastly, this last slide. I would like you to do the short test in Blackboard. So the test only has 5 multiple choice questions. It will help you to get familiar with the style of the questions asked in the exams and know how online exam works. The scores are not counted toward your course grade, so take advantage of it. One important thing to remember is that some of the questions in the exams are more difficult than these 5 questions. So please make sure that you watch and understand everything in my lectures and remember the key concept I emphasized during the lectures. Feel free to ask questions during the office hours through Zoom, okay? That's all for today. On Wednesday afternoon, we'll have this Q&A session, and please attend if you have any questions regarding anything about this course, all right? Thank you and see you next time.